Hi, we are Tomocube, and we believe imaging and analyzing cells in their native states are crucial. So, Tomocube develops and presents Holotomography HT2 and HTX1, which enable label-free 3D quantitative imaging of cells. Without introducing any exogenous labeling agents, it allows us to visualize the subcellular structures of live cells with unprecedented details and for a long-term investigation. It will enable various interesting investigations in microbiology. These are representative images taken with the Tomocube HT platform for the applications in microbiology. For example, you can image 3D shapes of Ralstonia and their internal structures over time. Yeasts can also be well imaged using HT, and you can watch the budding of yeasts. In the bottom left, you can see the individual Toxoplasma gondii parasites in the presence of potential host cells. The cell shapes and internal vacuolar structures of Candida albicans are quantitatively measured for systematic analysis. Important feature of HT is not just 3D label-free imaging. It also provides quantitative information such as bacterial cell mass, volume, and concentration. For quantitative analysis, we also provide an automatic bacteria analysis toolbox. It enables the seamless and automatic workflow, including the measurement, segmentation, and quantitative of various parameters of unlabeled live bacteria. HT2 reconstructs 3D distribution of the refractive index of individual bacteria with the resolution of 110 nanometers, from which quantification of the biomolecular concentration of each bacterium can be retrieved because there is a linear relationship between a biomolecular concentration and a refractive index. Because HT is a 3D imaging technique, the volume of each bacterium can also be calculated. From the measured concentration and volume of a bacteria, the dry mass of the bacteria can be obtained.
Exploiting such powerful imaging and analysis features, our users can perform systematic investigations of bacteria. For example, in a recent work, the growths of individual bacteria are measured using HT in the presence of antibiotics in various concentrations. The researchers have retrieved cellular volumes, dry masses, and concentrations. Conventional imaging approaches have several limitations for the study of bacteria. For example, for live cell imaging, one can use phase contrast or DIC microscopy. They are label-free techniques, so you can observe bacteria without treatments. However, they only provide qualitative information in 2D. Fluorescence microscopic techniques have been used due to their molecular specificity. But, you must follow several steps for cellular imaging, such as transfection for fluorescent protein expression or staining. It also requires washing, incubation, and fixation. These procedures are not only time-consuming but also inevitably cause phototoxicity or photobleaching. One crucial issue is that cells are significantly modified from their native states and should be avoided for long-term investigation. Furthermore, the obtained images are qualitative. Researchers do subjective assessments by observing cellular images represented with imaging contrasts with arbitrary units. To solve these challenges, Tomocube presents unique solutions based on holotomography, HT. HT measures the 3D tomography of live unlabeled cells within a few seconds. The reconstructed tomography has quantitative information refractive index distributions, which can be directly converted into biomolecular concentrations. Thus, one can also retrieve quantitative bioimaging information such as volumes, concentration, and dry mass of subcellular organelles.
Tomocube provides two product lines. HT2H is a high-resolution HT system well fit for small-size research labs. Recently released HTX1 is designed for imaging facilities and biotech companies. HTX1 provides high-through output analysis. It can measure cells and tissues in various carriers, such as imaging dishes or commercial multi-well plates. Both HT2H and HTX1 provide a feature for measuring 3D deconvolution fluorescence imaging with 3-channel overlay and an incubator for long-term measurements. There are interesting scientific papers produced using HT, particularly for the field of microbiology. For further information, please visit www.tomocube.com or contact us via info@tomocube.com. Thank you.